This entry for the UNDP COVID-19 Detect and Protect Challenge is a precision heat block for use in COVID-19 diagnostic testing with the next generation of COVID-19 tests, which use a technology called CRISPR. These tests are inexpensive, easy, and quick, but they do need a precision heat block for each of the two phases of the test reaction. There are two separate phases. Each phase requires its own temperature, and it requires that temperature to be controlled. This unit has heat blocks designed for 0.5 milliliter centrifuge tubes, but it could be easily changed for other configurations of the tests. The unit is designed to be used remotely, so it's independent of things like the power structure. It runs from any 12-volt supply, 12-volt DC, which can be a wall bug like the one shown, a power supply. It could be a car battery, or it could even be a solar panel, anything that can provide 12 volts DC at 2 amperes. Once plugged in, the unit immediately starts to heat the heat blocks to the correct temperature for each phase. The blue flashing LEDs indicate that the heat blocks are still heating up. They haven't reached the correct temperature yet. When they reach the correct temperature, they turn green. You can see the two phases of the reaction laid out, one called RT lamp, so reverse transcription and amplification of the RNA potentially from the virus. And the second phase, CRISPR, where we use CRISPR to actually detect whether there was viral content in the sample. So you can see that the RT lamp heat block has reached temperature. And what would happen is the reaction tubes are provided with the CRISPR diagnostic test, and they include a small pellet of freeze-dried chemicals that are used as part of the test. So in use, one puts a small amount of distilled water into the reaction tube and the sample into the reaction tube, caps it, inserts it in the RT lamp section of the heat block, and pushes the RT lamp timer. The timer now ticks off the time required for that portion of the test. It's about 20 minutes, so we'll shorten the video. We'll edit the video to take out the 20 minutes, but in the end, once the RT lamp timer has expired, the user would move the reaction tubes to the CRISPR heat block and start the CRISPR timer. When the CRISPR timer is done, the reactions are done and they're ready to be used with a lateral flow device, such as you find with a home pregnancy test, that kind of a paper strip. And you would put the reaction tube at room temperature, insert the lateral flow device, and let it sit for a few minutes while the solution crept up the lateral flow device to form lines that tell you either the virus is present or the virus is not present. So a quick, easy test enabled with this precision heat block that controls the temperature to the temperatures required for the reaction.